Welcome to this video. In this video, I will be talking about molecular biology and structural biology. So let's start with molecular biology. What exactly molecular biology is? So it is the branch of biology that deals with the structure and function of molecule which is essential to life, particularly DNA, RNA and protein. It focuses on how genes are regulated, how they function, and how these molecules interact to support cellular processes. Now, coming to techniques in molecular biology, there are several powerful techniques used in molecular biology to study these molecules. Let's take a look at some of the most common ones. PCR, polymerase chain reaction. This technique is used to amplify a specific DNA sequence, creating millions of copies of a target DNA segment in just a few hours, and it is a very essential tool for genetic research, diagnostic, and even forensic sciences. Now, gel electrophoresis. Gel electrophoresis separate DNA, RNA, or protein based on their size and charge, allowing scientists to analyze and compare molecular fragments. Molecular cloning. Cloning involves inserting a DNA fragment into a vector such as plasmid and then introducing it into a host cell. This allows for the production of large quantities of a specific gene or protein. In the last, there is CRISPR-Cas9. This is a groundbreaking gene editing technology that allows scientists to modify DNA with precision. It is used in gene therapy, agriculture, and even potential cures for genetic disorders. Overall, molecular biology techniques are very essential in understanding life's molecular foundation, from diagnosis diseases to discovering new treatments and advancing research in genetic engineering. The ability to manipulate DNA, RNA, and protein continues to push the boundaries of medicine and science, making it a vital field. Now, this is a picture of DNA we all know. DNA is deoxyribonucleic acid, a molecule that stores our genetic information. It is the blueprint for all living organisms and direct the synthesis of proteins, the molecules that perform most of the function in our cells. RNA and proteins. So, you know the central dogma from DNA to RNA to proteins. This is the central dogma of our life. So, RNA or ribonucleic acid is closely related to DNA. It plays several key roles, including carrying the instruction from the DNA to ribosome, where proteins are made. And after expressing the protein, our body performs its function, certain body parts, or RNA is also involved in gene regulation and expression. Now, coming to structural biology. So, this field is all about understanding the 3D structure of those biological molecules like proteins and nucleic acids. Why is this important to study? Because the shape of a molecule influences how it functions. By studying the structure, we can learn how these molecules work, interact, and even how to design drugs, medicines to target them. And these are the techniques in structural biology. Several techniques are used to study the structural biology molecules, including circular dichroism. Circular dichroism, a molecule absorbs circularly polarized light, providing information about its secondary structure, such as alpha helices and beta sheets in proteins. Now, biosex. Biosex is another technique which helps to determine the structure of any protein or complex with DNA. Cryo-EM. Cryo-EM freezes samples at extremely low temperature to capture high resolution images of biomolecules in their native state. It is a cutting edge method for visualizing complex structures. NMR spectroscopy. NMR or nuclear magnetic resonance allows us to determine the structure and dynamics of molecules in solution by observing the magnetic properties of atoms within the molecule. X-ray crystallography. X-ray crystallography involves shooting X-rays at a crystallized molecule to determine its structure based on the diffraction pattern. It's been crucial for many groundbreaking discoveries, including the structure of DNA. I hope you liked the video. Thank you for watching and please subscribe for more.